Hi, my name is Melanie Föll. I'm a postdoc in Olga Witek's lab at Northeastern University in Boston. I will present mass spectrometry imaging in the age of reproducible medical sciences. The work I present here has been published in GigaScience last December and was part of my PhD thesis, which I performed in the lab of Oliver Schilling at the Institute for Surgical Pathology in Freiburg. Mass spectrometry imaging, short MSI, is able to reveal the distribution of thousands of analytes such as metabolites, lipids, peptides, proteins and drugs. Therefore, MSI is used in different research areas, including preclinical and clinical research. In pharmacological studies, imaging of drugs and their metabolites in animal models allows the investigation of the drugs' kinetics and toxicology. MSI is often used to study solid tumors because they are spatially heterogeneous and can be virtually dissected by MSI, as seen in this colorectal liver metastasis. A common application for MSI is the discovery of potential biomarkers for diagnostic assays or the prediction of outcome or therapy response. MSI can be performed with different types of mass spectrometers. The most common are multi-TOF devices. The multi-laser scans over thousands of tissue locations in a grid with a spatial resolution in the lower micrometer range. In each location, a full mass spectrum containing thousands of analytes is recorded. The hyperspectral data is later assembled into distribution profiles for each analyte. Those profiles can be compared with the optical image of the sample and used for further statistical analysis. MSI data analysis remains challenging due to the large and complex data, missing standardization and accessible software. In most studies, proprietary or license-requiring software as well as in-house scripts are used for the analysis, which hinders accessibility and reproducibility. Existing open-source software that paves the way for reproducible data analysis often requires steep learning curves for scientists without programming knowledge and sometimes lengthy installation of software. To overcome these problems with MSI data analysis, we would like to introduce the MSI community to the Galaxy framework. Galaxy is an open-source data analysis platform that is easily accessible via a web browser and a graphical user interface that allows the analysis of pre-installed tools on large computational resources via public clouds. Galaxy sets high standards in terms of reproducibility. The analysis history keeps for each analysis step the name of the tool version, and all specified parameters. Tools can be combined into workflows that allow high levels of standardization and automation. Furthermore, workflows and analysis histories can be shared privately with colleagues or publicly, for example, as part of a publication. To build MSI tools in Galaxy, we have integrated a variety of existing open-source algorithms. Most functionalities are based on algorithms of the versatile MSI software Cardinal. Multi-quant MS preprocessing algorithms were used, as well as scikit-image as basis for the development of image co-registration and regions of interest annotation functionalities. For each software, we have built conda packages and containers to allow automatic installation of all dependencies and version control of the MSI Galaxy tools. For each Galaxy tool, a wrapper was created to define inputs, outputs, and parameters to build a graphical user interface. All MSI tools are installed on the European Galaxy server and available in the Galaxy toolshed as well as a Docker container. On usegalaxy.eu, our tools can be run on public clouds that provide about 6,000 cores and 28 TB of RAM. In addition to the MSI tools, there are about 2,500 pre-installed tools on the European Galaxy server that can potentially be included into the MSI analysis, for example, tools for image analysis, machine learning, and classical proteomics and metabolomics analysis. For sensitive data, the MSI Galaxy Docker container can be installed on any type of computer to allow the analysis in a closed network setting. We split up the different analysis functionalities into 18 Galaxy MSI tools that can be combined in a user-defined manner and allow all major MSI analysis steps, including quality control, data handling, pre-processing, co-registration and region of interest annotation, supervised and unsupervised statistical analysis, visualization, and identification. One highlight is the automated quality report, which contains more than 30 descriptive statistical plots that allow to access, for example, spatial gradients, batch effects, and mass accuracy. Another highlight is the availability of more than 30 different pre-processing algorithms. To help users getting started with the Galaxy MSI tools, we have built 3 hands-on tutorials that allow the analysis of small but meaningful proteomics and metabolomics datasets. We applied the Galaxy MSI tools to reanalyze a publicly available dataset of N-linked glycans imaged in mouse kidney tissues. We obtained similar results as the original study and made the analysis history and the corresponding workflow publicly available to highlight Galaxy's reproducibility and transparency. Currently, we use the MSI Galaxy tools to analyze peptide distributions in a variety of cancer tissues. 2 examples include a study of a patient cohort with different types of bladder cancer, and another study compares matched primary tumors and liver metastases in colorectal cancer patients. In the bladder tissue cohort, we found discriminative peptide features that distinguish tumor and stroma histology, and another set of peptide features distinguished invasive versus non-invasive carcinomas. For more information on the bladder cancer study, please visit my poster. I would like to thank all colleagues from the Schilling Lab and from the VTech Lab, as well as all collaborators from the Institute for Surgical Pathology in Freiburg, from the BioQuant Institute in Heidelberg, and the Galaxy Team Freiburg.